I'm Shelby Hogan, and I am doing an honors option for MMG 201. My honors option topic was microbes communicating via quorum sensing. To start off, what is quorum sensing? Quorum sensing is known as the regulation of gene expression in response to variations in cell population density. Microbes that are quorum sensing produce and release molecules called chemical signals. These chemical signals are also known as autoinducers. Autoinducers increase in concentration as a function of cell population density. Autoinducers can lead to an alternation in gene expression when there is a minimal threshold stimulatory concentration detected. Continuing with what quorum sensing is, there is synthesis of autoinducers by the bacteria or microbe. Once they are synthesized, they are then recognized, and once they are recognized, they are then responded to by the bacteria or microbe. The response can be things such as virulence, bioluminescence, or other group behaviors that the microbes or bacteria have to respond to when the host has been compromised. Autoinducers. There are different autoinducers when different things are happening. For example, gram-positive bacteria have different autoinducers than gram-negative bacteria. Gram-positive autoinducers are called oligopeptides. Oligopeptides have a very unique chemical structure that looks like this, with these two R groups, R and R prime, being different carbon groups that can be connected to the molecule. Gram-negative bacteria have another special autoinducer called N-acyl homoserine lactone, AHL for short. This molecule looks like this, also unique and different from the gram-positive bacteria autoinducer. There is also an R group, which also represents different carbon groups that can be connected to the molecule. Now, a third group of autoinducers are interspecies autoinducers, which have two types, V and S. These two types are both unique in their own ways, but one thing they have in common is that they have a lot of alcohols connected to them, and they have 3D structures, meaning these molecules are more 3D compared to gram-positive and gram-negative molecules, which are not, and meaning they also have stereochem. Gram-positive and gram-negative bacteria. Gram-positive and gram-negative bacteria use quorum sensing methods to regulate many different physiological activities. These physiological activities may include things like symbiosis, virulence, competence, conjugation, antibiotic production, motility, sporulation, and biofilm formation. Some of these most important ones in quorum sensing include virulence and biofilm formation. Gram-negative and gram-positive bacteria can be detected in two different ways. First of all, gram-positive bacteria are always pink. So you know when it is gram-positive because the bacteria will be pink when you look at it under a microscope. The gram-negative will always be purple. Although they are both cocci and bacilli, they are two different things, and you can tell by the pink and purple colors. So why do microbes communicate via quorum sensing? Well, quorum sensing allows microbes to communicate back and forth and coordinate their behavior. One example is because of environmental changes. Environmental conditions change very fast, and microbes have to respond right away. Some of the responses they have include adapting to nutritional availability, defense against microorganisms, and avoidance of toxic dangerous compounds. It is very important for microbes like pathogenic bacteria to co-coordinate virulence during infection of a host so that they can try to escape the immune response of the host. That way, the bacteria can establish an actual infection. So, opposite of what we as the host want, but exactly what the microbes and bacteria want. Bioluminescence. Bioluminescence is something that can be a response of the autoinducer's attachment. So, LuxI synthesizes acylated homoserine lactone AHL autoinducers. The AHL autoinducers freely diffuse across the cell membrane. When cell density is high, the autoinducer concentration increases in the cell's environment. This then increases the number of bindings of AHL to LuxR. LuxR AHL acts as a transcriptional activator of leucophorase genes. The expression of leucophorase results in the bioluminescence. Quorum sensing also has effects on ecology and evolution. Microbes gain strength in numbers by secreting virulence factors required for pathogenesis together as a unit. Throughout quorum sensing, microbes work together in order to gain benefits that are specific to their group. Example, altruism. This is when part of the population sacrifices one thing or is unselfish so that the majority of the population can survive and evolve. These are my sources, and that is all I have to say about quorum sensing and how microbes communicate.